Thank you for joining us. We are going to be handling cytology or cell biology. And by definition, cytology is just the study of structure and function of cells of plants and animals. But we are going to be interested much now on cell organelles. By definition, a cell organelle is a separate structure within a cell which performs a specific function. For example, the mitochondrial, the chloroplast, nucleus, and many others. And here we are going to start with the nucleus. As you can see, we are saying the nucleus is the most largest organelle among all organelles in eukaryotic cells. For this reason, we say it to be the most prominent feature of the cell. Its size and position varies. For example, it varies from cell to cell. For example, in plant cells, it is found at sides, and in animal cells, it is found in the middle. Those you can remember from O level the structures we used to develop a plant cell of animal cell. You can remember that. You can see here the nucleus for animal cells just situated in the middle. And then for the plant cells, the nucleus at sides. We are going to be describing the structure. Now, we're going to describe the structure of the nucleus. From my simple structure here, my drawing, it's always good when you're describing these structures. For all cell organelles, you start from outside going inside. As you can see, we're going now to start describing. This is the simple structure of the nucleus. We're going to describe it from outside inside. So it's bounded by a double membrane called the nuclear envelope. As you can see, this is the nuclear envelope. This nuclear envelope consists of two membranes, that is, say, the outer membrane and the inner membrane. As you can see, this is the outer membrane, this is the inner membrane. The outer membrane is continuous with the rough endoplasmic reticulum and often having ribosomes on its surface, and therefore it's rough. This is the outer membrane. It has ribosomes, which makes it to be rough, and is continuous with the endoplasmic reticulum. The inner membrane lacks ribosomes, and therefore it is smooth. This is the inner membrane. You can see it lacks ribosomes. It makes it to be smooth. The nuclear envelope consists of large pores, typically 3,000 per nucleus of usually 50 micrometers in diameter, which permits the passage of materials in and out of the nucleus. For example, the RNA, newly synthesized ribosomes, proteins, and lipids. It's always good when you are stating any structure, as in the organelle, you attach it with its importance. We said it consists of the nuclear pore. As you can see, this is the nuclear pore. These ones, and we have stated their importance. And now we are entering. We have finished this one, the nuclear pore. Now we are going inside. And then the inner membrane, as you can see, encloses a cytoplasmic-like fluid called the nucleoplasm. This is the nucleoplasm, the fluid inside here. It's what we call the nucleoplasm. But this nucleoplasm, now what is inside the nucleoplasm? The nucleoplasm contains a spherical body called the nucleolus, which aids in the manufacture of ribosomes and ribosomal RNA. As we said, when you state a structure in the organelle, you state it with its function. Now this is the nucleolus, as you can see. Then the nucleoplasm, as you can see, also contains chromatins. These are what we call the chromatins. You can see this is a chromatin. Chromatins, this one and this one. Specifically, the chromatin is specifically the heterochromatin, but this heterochromatin appear as dark, tightly coiled patches. Tightly coiled patches when observed in the electron microscope, and chromatins which appear as loosely coiled structures. These are the chromatins, and these are the heterochromatin, making the chromatin. We are saying specifically heterochromatin and chromatin because there are other chromatins. For example, the perinuclear chromatin, the internuclear chromatin, those are also found in a cell, but specifically the chromatins and the heterochromatins. So that is the structure. You start from outside going inside. That's the structure of the nucleus. So we can state some functions of the nucleus. Yeah, and the adaptations. Some the functions, you can see it controls cell activities. For example, the metabolic activities. It contains the genetic material of the cell, that is the DNA. It directs the process of protein synthesis. Yeah, it is involved in production of ribosomes and ribosomal RNA. And then we go for actually adaptations of the nucleus to those functions. When you are getting the adaptation, it's just simple. You set the function, actually you set the function and the structure which do that, then you can make an adaptation. For example, the DNA is long to store many genes. The nuclear membrane is perforated, as we said when you are describing the structure, is perforated for exchange of DNA and RNA between the nucleus and the cytoplasm. Then presence of nucleolus enables the production of ribosomes, which are protein factories. Now satisfying this function that it directs the process of protein synthesis due to what? Presence of the nucleolus. And then the nuclear envelope isolates nucleus from interference by processes in the cytoplasm. The nuclear envelope from the cytoplasm. Remember, these structures, the nucleus is also found in the cytoplasm of the cell. So the nuclear envelope just isolates the nucleus from interference by the process in the cytoplasm. Yeah. Okay. Then the nuclear pores are narrow, therefore regulating entry and exit of materials. So that is the structure of the nucleus, its function and adaptation.